Laughter in science is a rare phenomenon. However, the Ig Nobel Prizes concentrate on humorous research and give us the opportunity to have a good laugh. Founded in 1991, the Ig Nobel or Schnobel Awards are a good parody of the Nobel Prize. They are awarded for achievements that first make people laugh, then make them think. The award ceremony is filled with lively events, including mini-operas, scientific demonstrations, and so-called 24/7 lectures, where scientists explain their research twice, once in 24 seconds and the second time in just 7 words. The winners' speeches are limited to 60 seconds. And although the awarded work may seem funny at first glance, this does not mean it lacks scientific value. So the nomination for Physiology. You might remember the children's joke about a hedgehog that learned to breathe through its butt, only to sit on a stump and suffocate. A team of Japanese scientists led by Ryo Okabe, likely familiar with this joke, decided to investigate whether mammals could breathe through their anus, just as some fish and sea cucumbers do. It all began during the COVID-19 pandemic, when there was an acute shortage of ventilators. In search of alternative methods, the scientists turned their attention to the modest loach, a fish that can breathe through its rear end thanks to the large number of capillaries in its intestines. They thought, why not try this on mammals? And guess what? They indeed conducted experiments on mice and micropigs, introducing oxygen, yes, anally. The results were impressive. This method of breathing can indeed prevent respiratory failure without serious complications. Who would have thought that oxygen enemas could be so effective? This discovery could open new horizons in medicine and help patients in need of ventilation support.
You should have long learned to breathe through your butt. I see you're starting to do this after sniffing your slippers, which I had previously peed on.
The next nomination is peace. What peace can exist without rockets, right? The story began in the early 40s of the 20th century. The Americans came up with a rocket called the Pelican. The rocket was so packed with explosives that there was no room left for large guidance systems. Then American psychologist B.F. Skinner proposed using pigeons to guide the rockets. Yes, pigeons. At a time when the world was on the brink of disaster, Skinner believed that ethical questions, like the use of animals in warfare, were a luxury of peacetime. Skinner noticed that pigeons were good at recognizing patterns and could accurately respond to visual images, so he decided to train them to aim at specific targets. The pigeons were placed in a special harness above a screen where they pecked at images of targets projected onto a transparent plate. It was something like a camera obscura. To increase the system's reliability, they decided to use not one but 3 birds, just in case one got distracted at a crucial moment. However, despite impressive results, Skinner faced skepticism. He once even joked that it's easier to train a pigeon than a physicist on the committee. Although the pigeon experiment was successful, the committee of scientists observing the demonstration couldn't take the idea of a live bird guiding a missile seriously. It seemed too fantastical, and instead of excitement, there were more restrained smiles. Despite the fact that the pigeon guidance system didn't require rare resources, was simple to manufacture, and could be mass-produced in just 30 days, the committee rejected the idea. Military attention was already focused on the Manhattan Project, and Skinner was left with equipment that suddenly became obsolete, and several dozen pigeons who had become quite the geography experts of the New Jersey coastline. Yet in the 50s, the project was briefly revived under the name Orcon, and although pigeons were no longer used, this idea helped in the development of new radar technologies. Skinner never lost faith in his crazy ideas, and perhaps thanks in part to such experiments, science occasionally takes unexpected steps forward.
It's a pity they didn't continue the research. A dove of peace guiding a nuclear missile. That would have been a genius symbol.
Nomination physics. Rather unexpected experiments for physics. James Liao, a physicist studying fish hydrodynamics, has been interested for many years in how fish can reduce energy costs while swimming. Liao and his team hypothesized that fish can use vortex flows in the water to reduce their resistance and literally go with the flow without expending their own energy. The vortices that form behind other objects can serve as an energy source, allowing fish to move more efficiently in the water. This concept is reminiscent of a kind of surfing on water currents. To test this hypothesis, Liao and his colleagues chose rainbow trout, a fish known for its ability to minimize energy costs during swimming. Liao's previous research showed that trout adjust their movements to synchronize with the vortex flows in the water, allowing them to swim more efficiently. However, the most interesting part of the experiment was using a dead trout to prove that even without active effort, the fish could swim using the energy of the currents. The scientists placed a dead fish in a vortex stream and were surprised to observe it starting to move, demonstrating movements similar to those a live fish would make using the energy of the currents. As Liao himself noted, the dead fish continued to swim upstream thanks to the vortices, although it could no longer stop. The researchers also conducted similar experiments with solid foil materials to confirm their results. The experiments showed that objects can use the energy of currents to overcome their own resistance without expending any energy of their own. This experiment not only provided new insights into how fish conserve energy, but also opened new perspectives for research in hydrodynamics and energy.
A trout with foil by the stream. It seems like these experiments were conducted at some wild barbecue.
Next nomination, anatomy. Could the direction of hair whorls on a person's head depend on which hemisphere they live in? This seemingly frivolous question intrigued a group of scientists led by Marjolaine Willems. Their study aimed to identify a possible connection between the direction of hair whorls, clockwise or counterclockwise, and geographic location, namely, whether there is a difference between residents of the northern and southern hemispheres. Hair whorls have long attracted researchers' attention. Some hypotheses even suggested links between them and certain physical or cognitive traits, such as being right or left-handed, or even sexual orientation. However, these hypotheses were debunked. Willems and her team decided to focus on another question: how geographic factors might influence the direction of hair whorls. The researchers conducted a retrospective study of 3 groups of children: the general population from the northern hemisphere, Paris, France; the general population from the southern hemisphere, Santiago, Chile; and a group of identical twins born in Paris. The results showed that twins often had hair whorls that spun in the same direction, indicating a significant genetic influence. However, among children from the southern hemisphere, the whorls were more often counterclockwise, suggesting a possible environmental influence, although genetic characteristics of the specific population could not be ruled out. Although the scientists even suggested the possibility of the Coriolis effect, the same one that, according to myth, causes water in sinks to rotate in different directions depending on the hemisphere, this assertion has long been debunked. Willems and her colleagues nevertheless left the possibility of further research on this issue. At the same time, they acknowledged that there is unlikely a direct connection between the direction of hair whorls and geographic conditions. Thus, although the idea of hair whorls as a geographic phenomenon may seem fantastical, this research raised interesting questions about the interaction of hereditary and environmental factors in the human body.
I believe the hardest challenge for the scientists was keeping a straight face during these experiments.
Next nomination, demography. Human mortality is an inevitable reality, but there are always a few people who seem to defy the laws of nature and live beyond 100 years. Scientists have long been interested in such people, particularly their concentration in so-called blue zones, geographic regions where people typically live longer. Among the common explanations for this phenomenon are a healthy vegetable-rich diet, strong social connections, and certain genetic traits. However, Saul Justin Newman took a more critical approach to this topic, noting strange patterns. He observed that longevity in some blue zones was associated not so much with favorable living conditions, but with factors such as poverty, low income per capita, and even higher crime rates. This contradicted established views, and Newman decided to investigate the demographic data more closely. The results of his investigation were astonishing. In all the blue zones he analyzed, numerous errors were found in the records of births and deaths. For example, in 1997 in Italy, about 30,000 people were still receiving pensions despite having long been dead. In Costa Rica, the 2000 census revealed that 42% of citizens over the age of 99 had incorrectly reported their age. This significantly reduced the number of centenarians in the region and led to a decrease in average life expectancy. And in 2010 in Japan, more than 230,000 people who were considered centenarians were missing, fictitious, or long dead, accounting for 82% of record errors. Newman noted that in any other scientific field, the discovery of such a number of false data would cause a major scandal. However, in demography, these errors seem to go unnoticed. His research raises the question of whether blue zones are truly hubs of extraordinary longevity or just the result of poor record keeping.
That's exactly what I'm talking about. When boasting about your manhood, beware of the person with a ruler.
Next nomination, medicine. We are all familiar with the long lists of possible side effects that accompany medication instructions. It would seem that the best medicine is one that has no side effects, right? Especially considering the nocebo effect, where the expectation of side effects increases the likelihood of them occurring. However, Levin Schenk and his team decided to test an alternative assumption. What if even fake side effects could make treatment more effective? In their study, they focused on so-called active placebos, fake drugs that do not contain any active substances to treat the disease but cause noticeable side effects. The researchers hypothesized that mild side effects could strengthen patients' belief in the effectiveness of the treatment, thereby enhancing the placebo effect. To test this hypothesis, Schenk and his colleagues invited 77 volunteers to participate in the experiment. The participants were divided into two groups and were told they would receive a nasal spray with fentanyl, after which they would be subjected to heat pain using a special device. They were also told that the spray might cause mild burning in the nose as a side effect. In reality, neither of the sprays contained fentanyl. One group received a neutral spray with no side effects, while the other group received a spray with capsaicin, which caused a mild burning sensation, thus simulating a side effect. The results were striking. Participants who received the spray with the fake side effect reported significantly less pain than those who received the neutral spray. In other words, people felt more pain relief due to the expectation of side effects and the belief that the spray worked. This study showed that even fake drugs could be more effective if they cause mild side effects that reinforce a person's belief in the effectiveness of the treatment.
So what does this mean? Should we add a little laxative to homeopathic remedies? An extremely constructive idea.
Next nomination, botany. It turns out that some plants may be much more attentive than we thought. In 2013, botanists discovered an unusual plant in the tropical forests of southern Chile, the woody vine Botany trifoliolata. This plant has an impressive ability to mimic the leaves of up to 3 different host plants. Moreover, it not only replicates the shape of the leaves, but also their color, orientation, and vein patterns. This ability raised many questions. How does B. trifoliolata adapt so skillfully to its neighbors? There were several possible explanations. One hypothesis suggested that the plant receives information through chemical signals emitted by host plants. Another hypothesis proposed horizontal gene transfer between B. trifoliolata and the host plant. But what if plants have the ability to see? Jacob White and Felipe Yamashita decided to test this idea by conducting experiments. They took the Botany trifoliolata vine and placed it next to artificial plastic plants, including an imitation of a wisteria vine. The results of the experiment surprised everyone. B. trifoliolata began to mimic the shape of the artificial plant's leaves, even without direct contact with them. This discovery cast doubt on the hypotheses of chemical signaling or gene transfer, since plastic plants do not emit chemicals and cannot exchange genetic information. Thus, the researchers suggested that the phenomenon could be explained by a form of vision in plants, a system that allows them to visually perceive their environment. This finding opens new horizons for research. If plants are not only able to respond to chemical signals, but also to see or even perceive sounds, it could radically change our understanding of their capabilities and interactions with the environment. Perhaps we are still far from realizing the true complexity of our green neighbors' lives.
Now I understand. If there's a cactus next to your monitor, it's not a cactus. It just means the owner loves Pornhub.
Nomination probability. Flipping a coin is a classic way to settle disputes or make decisions, seemingly completely random. This is how it was decided which of the Wright brothers would fly first in 1903, or who would have the first pick in the 1979 National Basketball Association draft, which led to Magic Johnson going to the Los Angeles Lakers instead of the Chicago Bulls. However, physicists claim that flipping a coin is not entirely random, but rather deterministic according to Newtonian classical mechanics. The randomness arises from slight changes in initial conditions, such as starting position, flicking force, or angular velocity. It is widely accepted that the probability of heads or tails is equal 50-50, with no advantage for either side. But in 2007, Stanford statistician Percy Diaconis proposed that during a flip, the coin can slightly wobble. Its axis of rotation shifts direction in the air, which prolongs the time the coin stays with the initial side facing up. This creates a slight bias. The probability that the coin will land on the same side from which it was flipped slightly exceeds 50% and is 51%. František Bartoš and his colleagues decided to test this hypothesis. Although there had been many previous studies on coin flipping, starting with Count de Buffon's experiments in the 18th century and tests with 40,000 flips in 2009 designed to verify Diaconis's theory, the results were always inconclusive. Bartoš and his team surpassed previous attempts, collecting data from 350,757 flips, performed by 48 people. All but 3 were co-authors of the study. Each flip was recorded on video for greater accuracy. The results confirmed Diaconis's hypothesis. There is indeed a slight bias toward the coin landing on the same side it started from. However, no bias in favor of heads or tails was found. The researchers also suggested that future studies could check whether unstable flippers, those who make the coin wobble more during the flip, exhibit a greater bias compared to those who flip more steadily. However, they acknowledged that this would require significant effort, including a detailed analysis of high-resolution video for each flip. After all, even scientific curiosity has its limits for monotony.
Nothing surprising. Everything around has its habits. For example, a stone is used to lying at the bottom of a lake, not splashing near the surface. And even a blockhead like you has the habit of eating and breathing with your mouth, not like those fish from the Physiology nomination.
Nomination chemistry. Tess Harrimans and her colleagues decided to study the hydrodynamic properties of large active polymers, aiming to figure out how to separate these polymers based on their activity. Polymers are usually sorted by size, but the researchers were interested in whether it could be done based on their activity, that is, how much they move. For this, they needed samples that mimic the structure of large polymers, and nature already had a ready solution. Tubifex worms, multi-segmented creatures resembling long polymer chains. The team placed these worms in a specially designed channel, similar to those used in chromatography to sort polymers. But instead of polymers, there were worms. They were divided into two groups, active and inactive. To make one group of worms less active, they were soaked in a solution with ethanol, effectively getting them drunk. For convenience, a blue dye was added to the solution for the inactive worms, while the active ones remained red. The results were highly illustrative. The sober, active worms easily made their way through the channel and reached the end much faster than their drunken counterparts. This demonstrated that flow through a structured space could effectively sort active polymers, or worms, based on their activity and length. The experiment proved that chromatography is a reliable method of separating active polymers, even if for a time these polymers are turned into drunken worms.
I already knew, without chromatography, that if you don't come home on time on Friday, you come late and blue. What I didn't know is that you, worm, are used in experiments.
Final nomination, biology. In 1941, Fordyce Ely and William E. Peterson decided to investigate how cows release, or conversely, hold back milk during milking. It was known that some cows easily give milk, while others seemingly hold it back, which had long puzzled farmers. Ely and Peterson developed a rather unusual method to test the effect of fear on the process of milk release. They placed a cat on the cow's back while it was being milked by a mechanical machine. Then, every 10 seconds for 2 minutes, they popped paper bags near the animal. As it turned out, the paper bag explosions alone were enough to have an effect, so the cat was no longer needed, as the authors humorously noted. During these experiments, they also studied the effect of adrenaline on the cow's ability to release milk, injecting the hormone directly into the bloodstream. Ely and Peterson concluded that the process of milk letting is a conditioned reflex triggered by high intraglandular pressure due to the action of oxytocin in the blood. Oxytocin stimulates the contraction of alveolar and duct muscles, which releases the milk. The retention of milk, however, was linked to the effect of adrenaline, which prevented muscle contraction, thus stopping the buildup of high pressure in the glands. Perhaps the adrenaline injections alone could have provided all the necessary conclusions without the involvement of the cat or the paper bags. But where would all the scientific flair be then?
And after this, you still wonder why I pee in your slippers. I'm just milking from fear, you idiots.
Give your likes, dislikes, subscribe, feed Martin using the link below the video, and see you next time.